Greetings and welcome back to the course. So, what we're going to talk about here for the next couple of vignettes, probably like three or four vignettes, is a really cool topic which has to do with molecules as documents of evolutionary history. And my thought is to try to use the Galapagos as a lens to look upon these kinds of questions. So, what I'm going to do in this first vignette is give a little bit of a background. And what I mean by that is I'm going to try and go back into the 50s and 60s when we had a subject of genomics without genomes. And I personally really love that for reasons that I'm going to explain to you. And we'll use the example of hemoglobin as an example of using molecules as documents of evolutionary history. And then once we've put all that stuff in play, then what we will do is we'll turn to an analysis of some of the key organisms of interest in the Galapagos. So the first thing that I want to say is that DNA science has really provided all sorts of important insights into these Galapagos, what I call Galapagos superstars. And each of these is a picture that either I took myself or was with somebody who took them. The upper left, probably many of you will recognize, is the tracks that's left by a marine iguana. This was on either Isabella or Santa Cruz Island. I think it was on Isabella. And, I've always found the marine iguanas to be incredibly interesting. They're beautiful when they're swimming in the water. And when they're waddling along on the land, they produce these really, really neat tracks. Obviously, the Galapagos tortoises are iconic, and we will have a lot to say about the use of their mitochondrial DNA as a window onto their relatedness to each other. There's an adaptive radiation that took place on the Galapagos Islands, but also who the closest ancestor is on the South American continent. The upper right, obviously, probably the most famous of the organisms on the Galapagos are the finches, and you can see that finch near its nest. The bottom left is one that maybe is less well known, but the Galapagos lizard is also a really interesting adaptive radiation. That one is a picture that we took while spending about 25 or 30 minutes just watching this male and female going through a courting dance. The middle picture on the bottom, I will have something to say about this notion of heterochrony and how it is that out of the, if I remember correctly, something like 40 species of cormorants around the world, only one of them is flightless. And those you will see, for example, on the islands of Fernandina and Isabella. And finally, there's some snails, which are also well known. So what I want to do is I want to talk about the use of technologies to learn about the history of the world. And I want to start with astronomical examples. So I'm always very excited. I have to say that, my own entrance into science passed right through a door that was opened up by Galileo to me. And, at a very young age, I read The Starry Messenger, which is the manuscript that you see on the right. So in 1609, if I remember the dates correctly, over a 45 or so day period, Galileo was turning his so-called tube to the heavens, and basically he observed things that were all in a way inconsistent with the current thinking, which was the geocentric hypothesis about the organization of the celestial sphere. And what I mean by that is, just for example, he noticed that Venus has phases, a really subtle but beautiful effect, and an effect that we would expect actually would be related to a planet circling the sun. He observed sunspots, but perhaps most importantly, he observed the four moons of Jupiter that you see here, ones that you can observe yourself with binoculars. And if you look in the third book of the Principia by Newton, you'll see that he used Kepler's laws to understand the relationship between the semi-major axis and the period of the orbit of these moons of Jupiter. So that's great. What we see there is that there's this technology, the technology in this case being the telescope, and this technology transforms the way that we actually engage with what the world around us is like. But the bigger question of the history of the world actually takes shape in the form of the spectroscope being put on the back of this telescope. So what I'm going to do is read you this short piece that comes from Nature magazine from April of 1902. It's written in a very charming way. If we tried to write like this in the present day and age, the manuscript would be rejected, not only by the editors, but by our fellow scientists, and I object. I'm not a fan of that. Don't agree with it, but whatever. So this article is speaking of the physical laboratory in Heidelberg, and what it says is its small beginnings, meaning the labs, small beginnings in the middle of the last century are marked by the name of Kirchhoff scratched on the window of what is now the private room of the senior assistant. From this window, one may look out over the Rhine plain towards busy Mannheim, as Bunsen and Kirchhoff did one night when a fire was raging there, and they were able, by spectroscopic examination of the flames, to ascertain that barium and strontium were present in the burning mass. So, that's really interesting. The idea that one could do chemical identity of things that are in a fire at some 15 kilometers away. This picture that I show is one that I took from the hills around Heidelberg, looking out towards Mannheim, just kind of curious about the angle, how it would have been that they might have actually been able to see the fire, and I still haven't really fully worked that out. But the same window also looks across the Neckar to the Heiligenberg, along the slopes of which runs the Philosopher's Walk, the chief of the many paths among the wooded hills around the town which the two friends were wont to traverse in their daily constitutionals. So, if you go to Heidelberg, which is kind of a rite of passage for young backpackers, for example, right by the cathedral, you go down to the river, you cross it, there's a bridge, and right on the other side of the river, you'll see a sign that says Philosopher's Way in German. You can go up and get exactly the view that was their daily constitutional. Bunsen is known to have said that it was during such walks that his best ideas came to him. One day the thought occurred, if we could determine the nature of the substances burning at Mannheim, why should we not do the same with regard to the sun? But people would say we must have gone mad to dream of such a thing. All the world knows now what the result was, but it must have been a great moment when Kirchhoff could say, Bunsen, I have gone mad, and Bunsen, grasping what it all meant, replied, so have I, Kirchhoff. Now, what does this have to do with the history of the world? So, I always love to tell anybody that will listen that, when you fly to Los Angeles, I encourage you to get a right-hand window seat. And the reason is because as you're coming into the city, you'll be at about 7,000 feet when you pass Pasadena, and you'll be able to look off to the right, and you'll see the Mount Wilson Observatory. This is the Hooker telescope used by Edwin Hubble in order to discover the expanding universe. And, in a way, the telescope, with its spectroscope, is looking back in time and telling us about the expansion of the universe and so on. And so, I'm making the argument that the expansion in our technologies allows us to learn very interesting things about the world around us. Well, so many of us, when we think about the history of life, are used to contemplating fossils. And here I show a picture of Niels Shubin with perhaps, I think it's a replica of the fossil Tiktaalik, which is this transitional form from the Devonian, which occurred between 380, 360 million years ago as our ancestors emerged from the water. So this is sort of a hybrid organism. And of course, the use of fossils as documents of evolutionary history is classic and well known. But there's a parallel world that has to do with phylogenies and the history of life on Earth, which can be based upon the use of molecules. So I just wanted to remind everybody there's only one diagram on the Origin of Species by Darwin, and this is it. It's a diagram that basically shows his conception of what we would now call a phylogenetic tree. And here's what Darwin has to say about it. The green and budding twigs may represent existing species, and those produced during former years may represent the long succession of extinct species, the great tree of life, which fills with its dead and broken branches the crust of the earth and covers the earth with ever-branching and beautiful ramifications. So this idea of how would we develop what I'm going to refer to as tree thinking, and for the purposes here, the vignette being molecules as documents of evolutionary history, what I have in mind, this tree is meant to represent a succession of species over time. So, here Darwin again, he's so eminently quotable. Would it be too bold to imagine that in the great length of time since the earth began to exist, perhaps millions of ages before the commencement of the history of mankind, would it be too bold to imagine that all warm-blooded animals have arisen from one living filament? So, I'm always intrigued by the sort of classic ways of coming to terms with phylogeny and the classification of organisms. And I've also always been very excited about the fact that many scientists, when they're young, have gone off on voyages of discovery and voyages of isolation. And Linnaeus was no different than Humboldt and Darwin and Wallace and Hooker and Huxley and Charles Elton and many others who went off at a young age. Actually, Jacques Monod was part of one of the Elton expeditions where they go off into the world and they observe the world. And, Linnaeus, the harvest for him was a classification of organisms on the basis of their morphological features. And, I've always been amused very much, or at least as long as I've been aware of it, with his Linnaean system, the system of nature. And the part I wanted to call your attention to was the paradoxa, which you see here in the middle. I'm mentioning that just because I think it's fun to imagine a guy being told about unicorns and wondering about them because, the idea of a horse-like animal with one horn is not as strange as it may seem, actually. We know about an animal in Africa that has a single horn, and we know about in the ocean animals that have single horns that are quite long, the narwhal. So anyway, what my main point of all this was just to note that the fossil record, morphology, anatomy allows us to do classification, but really in the 2 meters of DNA in each of our 100 trillion cells, that's in a way a most exquisite kind of fossil. So, so I already noted that there's this exciting sense in which new technologies transform our understanding of the world around us and can be a window on the history of the world. And one of the fundamental tools of modern science is DNA sequencing. And I'm a real fan of this graph, which shows the number of nucleotides deposited on the NIH databases. And I just wanted to call your attention to, as of the end of last year, there's something on the order of 10 to the 17th nucleotides deposited on the NIH website. And just to put that in perspective, it's important to note that, if I think about the complete works of Shakespeare, it's 37 plays. The plays have something like, I would say, 30,000 words. There's 6 letters per word. That gives you about 6 million letters. So one bacterial genome corresponds to a complete works of Shakespeare. And so all the letters in this database correspond to more than 100 billion copies of Shakespeare. And I wanted to contrast that with the Library of Congress, which is something on the order of 50 million books, something like that. So we're going to ask ourselves, how can we read DNA in order to learn something about the history of life on our planet? And before engaging with that question, I just wanted to note that, we have this nucleotide language, but there's much to be desired about what it means to actually read it. So I'm imagining that some of you that are watching this yesterday in class when I gave this vignette, one of the students beautifully read me the version at the bottom. But, there's different, this particular paragraph is one that can move me to tears when read by David Attenborough, for example. But when it's couched in these characters, although beautiful to my eyes, it's literally meaningless. But when in English, what it says is, there is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. So, with that as my background, let's talk for a moment about the DNA time machine. So, this paper, as you can see, from 1965, is by Emil Zuckerkandl and Linus Pauling. It was written in a building, within a couple hundred meters of my office. And they make this really wonderful remark where they say, in macromolecules of these types, meaning proteins and nucleic acids, there is more history in the making and more history preserved than at any other single level of biological integration. I think an even better version of this insight comes from Crick, who in the 60s made this remark, biologists should realize that before long we shall have a subject which might be called protein taxonomy. Well, before long has already come and gone. We are living in that era. The study of the amino acid sequences of the proteins of an organism and the comparison of them between species, it can be argued that these sequences are the most delicate expression possible of the phenotype of an organism, and that vast amounts of evolutionary information may be hidden away within them. So that is my introduction to molecules as documents of evolutionary history. What I want to do now in the next vignette is I want to talk about genomics without genomes. How is it that such brilliant, clever people were able to do sequence comparison as an evolutionary tool, but in the absence of sequence information. What I'm going to do is I'll start out by telling you about this paper from Zucker, Candle, Jones, and Pauling, and then after that we'll talk about Alan Wilson, one of my heroes from UC Berkeley, who, along with Vincent Serach, did an analysis of the relatedness of humans and their primate ancestors. So that's where we are. I hope what you got out of this is just a sense of the reality of this notion of molecules as documents of evolutionary history to complement what we get out of the fossil record. And one of the best examples that I know of this has to do with the evolution of cetaceans. There's a beautiful and quite complete fossil record. We can look at the loss of toothlessness and enamel and things like that at the level of the fossil record, but something we can also see in the form of pseudogenes in the extant organisms. So we've got this notion of the molecules as documents of evolutionary history. Next, we'll bring it to bear on some specific case studies.